Vanakkam. Welcome to this course on classics in neuroscience. In this course, we have been looking at neural circuits of the spinal cord, retina and some invertebrate systems. In this particular video, we will look at the work of Keffler Hartline to improve our understanding of retinal processing within the brain. Hartline was interested in understanding how the retina processes visual information, and he suggested that we can use comparative physiology, that is study model systems at a lower level to improve our understanding of how the species at a higher level function. Of course, there will be disadvantages with this method, but there are also certain advantages with this method. He describes his philosophy in his later papers. I am going to read from that. He says that you see the application of broad principles to specific cases of such complexity is not easy. So, that is where comparative physiology can help. So, you can't take broad principles and study relatively complex systems. So, that is his view. He describes that in his later work in 1967. Hartline graduated from Johns Hopkins in 1927 and he joined Eldridge Reeves Johnson Foundation for Medical Physics at the University of Pennsylvania, working with Detlef Bronk, who along with Adrian was one of the neurophysiologists, major neurophysiologists of that time. So, he was inspired by the single fiber study of Adrian Bronk that he then pursued, which he later published as the paper on application of unitary analysis to receptors and neurons in the visual system. He took the model system, the horseshoe crab, this Limulus polyphemus, and he studied its function and how the visual system in the horseshoe crab worked. It turns out that this is a relatively simple model system. It turns out that there were these eccentric cells, the light sensitive cells that had receptors. These eccentric cells had receptors that were sensitive to light. If you stimulate these receptor cells by providing physiological stimulus, that is light, it activates the central eccentric central dendrite, the eccentric cell itself. Remember, this crab actually cannot see the details of the picture being presented. It can only distinguish between darkness and light. It can only distinguish between light and dark for visual responses. So, you are wondering really what are we going to study or what are we going to understand by studying this species. It turns out that there are quite a few advantages to studying this, because this model system actually provided the opportunity to study the optic nerve that could be dissected down to single fibers. So, I could record from single fibers using simple methods like string galvanometer, for example. So, you can record information or responses from single eccentric cells that respond to light, and you just attach this to galvanometer with the electrodes and you are going to be able to measure activation when light or darkness happens. So, it is not going to detect the features, but just the presence or absence of light, for example. Hartline identified many crucial features or properties of the visual response. He found that a wide range of luminosity was represented into or compressed into smaller ranges of frequencies of impulse discharges. So, you are going to have a large range of luminosity that is going to be represented in relatively small changes in frequencies. Also, changes in light intensity were represented more vigorously. The responses to changes were more vigorous than just the presence or absence of light itself. So, there was sensitivity to change. This has been shown in other receptor systems, for example, in the muscle spindle itself and in other receptor systems also. That means that processing of the visual sensory information begins in the receptor itself, in the sensor itself. It is not merely passing on the information, but sends to the brain, whatever is the brain in that species, a form of processed information. So, the processing of these sensory signals first begins in the receptor itself. He showed this also in the frog optic nerve. Of course, an interest is to compare what happens when you have contrast, when you are having a single spot of light and when you change that to bands, for example, what happens, right. What Hartline found was that a single spot of light stimulated a single receptor and the nerve response was graded in frequency. So, there is more luminosity, there is a increase in frequency, for example. But suppose you had a dark and light region that passes through the site of the recorded cell. When you do that, there is on the light side a higher response and on the dark side a lower response, as you would expect. But what he also found was that there were inhibitory connections between eccentric cells such that those cells that were more strongly stimulated had less inhibition and their weaker neighbors had more inhibition. Thus, there was an enhancement of contrast between light and dark sites. So, there are two levels of processing that we are now speaking about. First is that changes in the intensity of the light causes more changes in response when compared with the intensity itself, when compared with the light itself, presence or absence of the light itself. That means there is more sensitivity to change, suggesting a first line of processing. But more than that, inhibition and excitation at the next level was such that the more stimulated cells had less inhibition and the less stimulated cell has more inhibition, suggesting a form of contrast inhibition. So, contrast inhibition is not necessarily happening at a deeper level, but at the second level, for example. So, there is this inhibitory connections between the receptor cells, the eccentric cells, are perhaps one of the reasons for this contrast enhancement that was first suggested by Ernst Mach. With this, we come to the end of this video. In this video, we looked at the work of Keffler Hartline to improve our understanding of the visual system. He used the crab visual system and how contrast enhancement was perhaps explainable using the inhibitory and excitatory connections between eccentric cells, at least in this model system, that is the limulus crab. Thank you for your attention.